Today I'm going to talk about microbiology of dental caries and periodontal disease. But uh, the topics actually is about oral microbiome. So first of all, I'd like to introduce you some overview information about organism and oral microbiome. The human mouth is heavily colonized by microorganisms. Those include virus, protozoa, fungi, and archaea, as well as the bacteria. So it includes several microorganisms. When focused on the bacteria only because it's a very large population of microorganisms present in the oral cavity. Actually, it's highly complex with more than 700 and up to 1,000 species of the bacteria in the mouth. At the oral site, it is the second most complex after the colon. So when we try to look at the big picture or entire our human body, we can call a collection of organisms. In this case, we focus on microorganism. We call human microbiome. This means that we talk about the entire human body. It can be used as microbiome or microbiota, but actually it is different between these two terms, two keywords. Microbiome, it might show a broad or bigger picture than microbiota. So it depends on the site of the human body. Maybe gut microbiome, oral microbiome. But at the oral microbiome, it is the subset of the gut microbiome. Gut, it means from mouth to anus, or our digestive tract start from the oral cavity and then in at the anus. But in our body, it has other sites. For example, it may be skin microbiome. It may be stomach microbiome or maybe urogenital tract microbiome. Okay, so it depends on the site we are going to study or going to analyze it. Okay, go back. In order to try to observe or in order to identify the bacteria, how many of the bacteria, how many species, and in order to observe the diversity of the bacteria or microorganism, we have to use the technique that to identify them. We can use several techniques, but the conventional techniques we use culture. We have to culture the bacteria and then have to identify it. The first method, culturable, use the technique that have to grow it. Or we can use other technique that can be used for a group of bacteria that could not culturable. Include the technique that can pick up the species that are unculturable. This means it could not be grown because the condition for growing of the bacteria is very unique. For example, you have to grow it during oxygen-free condition. It means it could not be grown normally. You have to use the room that have special condition. I mean, special with other condition without oxygen to grow it. This means that it could not grow normally at normal condition. So actually, now we have the technique that can pick up both culturable and unculturable. So you will see later. The two commonest diseases are caused by bacteria, dental caries and periodontal disease. Dental caries process was mediated by microorganisms capable of producing acid and digesting protein. Acid, the main factor that destroy the teeth, and some kind of the protein that can able to digest might be digest the gum, the junction between the gum and the teeth, or it can be digest enamel. So these protein might be produced from the bacteria. So you will see the term mutual interaction of multiple bacterial species. I mean, actually, in the oral cavity, it has several species of bacteria, but they live in peace. It means it can help each other to survive in the condition of the oral cavity. So we call the interaction that help each other to survive, we call mutual interaction. A group of streptococcus, sometimes we call mutants, streptococcus or streptococcus mutans. These bacteria was found to be carcinogenic bacteria, but they found in the test, it has test in the experiment in the animal. So it seems that this bacteria might be present in the oral cavity and destroy some mutual interaction. So this species might be dominant to over other species. Okay, go back to the techniques. The modern techniques we call the technique that used to determine the composition of the bacteria is mouse. We call culture independent method. This technique, it includes several approaches. We use phylogenetics, metagenomics, transcriptomics, proteomics, and metabolomic approach. So it has several levels. So we use several approaches to dissect, I mean to look at more detail on oral host microbiome interaction relevant to health and disease. So start with the first one, composition of oral microbiome. Composition of oral microbiome. So if it's microbiome, it means all microorganism. Microorganism is include virus, bacteria, fungi, but also archaea, as I mentioned before. So the virus, what kind of virus we found? Most of the virus found is disease-associated virus. It might be the mumps, rabies virus, that might be present on the cells expressed or produced from salivary gland. Hepatitis virus and HIV versus the virus, but these virus are blood-borne virus. This might be released from the gum disease or might be breathing on the gum. Herpes simplex, especially herpes labialis, the herpes on the lip, on the mouth, or human papilloma virus or the skin virus, bacteriophage, or the virus or bacteria. So it has several viruses, both disease associated virus. If they are not disease associated, it might be present because it might be eliminated from the human immune system. The second one, protozoa. Protozoa, example of protozoa might be Entamoeba gingivalis. The protozoa that's present in the gum, or Trichomonas tenax. So the population of these protozoa increased among poor oral hygiene and gingival disease persons. Potential pathogen, but currently regarded as harmless saprophytes. I mean, sometimes we thought it, previously we thought it might be pathogen, but later we found that they are harmless saprophytes or scavenger or destroyer, I mean destroy the babies. So the meaning of poor oral hygiene, which means there are a lot of food debris or bacteria in the mouth because the protozoa eat bacteria. So protozoa eat bacteria. The third group, or the fungi group might be Candida species, but we normally know that these Candida are symptomatics or the normal flora. From the literature, we found that almost 85 genera found in the healthy individuals. This from the research. In healthy individual, we found it might be more than 85 genus. So predominant genera includes Candida, Cladosporium, and so on. Other possible, sometimes we found that it might be contaminated from air or from food because you ingest the food by using mouth. So the debris present debris from outside the body, and sometimes it contaminates. Okay, move to number 4, archaea. Actually, archaea are different from bacteria as we know previously from the biology class. Archaea account for a minor component of the microbiome in the mouth. It can be detected in a healthy individual, but it will increase in the periodontitis person. Both of them are methane producer, so you will see the species name. It will start with methanol something. For example, methanol Bivio Bivibacter olaris, methanobacterium carbon, for example. Okay, move to the last group of the microorganism found in oral cavity, the bacteria. If you use the conventional method or microscopic method to see to see morphology of the bacteria. This method is very primitive. This is the first method that we use to collect the species of bacteria found in the dental plaque. And sometimes we can culture or grow the bacteria, we call the notch method. But later, Pasteur and others try to collect and classify the bacteria. The bacteria found in the oral cavity can be classified into two different groups. The first one is aerobe. This aerobe means it loves oxygen, and the second one a group of anaerobe. The anaerobe is hate oxygen. So the meaning of love and hate is mean that they can survive in the environment with and without oxygen. So we have to think about it seems that our oral cavity it should have oxygen, but sometimes in some in the cavity or in the area between teeth, sometimes it lack of oxygen. So some bacterial species can grow in that area, and some bacteria they have ability to trap other bacteria and hidden itself in between other bacteria, and the condition where they hidden in that area it seems to be anaerobic condition. Okay, most of the bacteria they degrade simple or complex carbohydrate or complex sugar as well as complex protein. Sometimes we found that administration of the antibiotics can alter or change the view of bacterial composition in the oral cavities. It seems that if you use only culturable technique, we see the distort view of the bacteria. Distort view is mean the view that is not clear. It might be you might see the different information from the fact. So we have to use the other technique. Okay, move to this slide. This slide try to explain the way we observe oral microbiome. So the whole oral microbiome are shown in the blue green area. But this area include both unculturable and culturable microbes. The conventional method that previously used can detect only the culturable microbe, but it could not detect the unculturable microbe. But the new technique from now, sometimes we call metagenomic technique, it can be detect both unculturable and culturable microbe. So you have to know about this. The meaning of the metagenomic technique is better. It is better than the conventional method. Okay, move to the part of technique that used for microbiome composition analysis. In order to study the microbe or microbiome composition, we have to use the molecular technique or the conventional techniques. So from this table, you should classify the difference between the metagenomic techniques as I previously mentioned before, and you have to compare some some procedure and the purpose and advantage and disadvantage of two type of techniques, the metagenomic technique and conventional microbiology. Okay, try to study and remember the name of the technique. Okay, go back to the oral microbiome. Actually, the meaning of the oral microbiome is a collection of all microorganisms. So, in order to know or every organism found in the oral cavity, we have to use the metagenomic technique to observe and analyze the composition of the uh, microorganism found in the oral cavity. Actually, in your oral cavity, it has three different bacterial community sites. The first site, buccal mucosa, gingiva, and hard plate. It seems that the different are not present in this location. So it means that every person, every area from this number one have the similar pattern of the bacteria. But the second one and the third one, second one, saliva, tongue, toxin, and throat, it have different bacterial community. And the third, supra and subgingival plaque, it also have the different diversity of the bacteria. When compare between each individual, it seems that we share 47% of species. It means we have 40% of species that are the same species, but other, the rest, approximately the 53% are different. So we call the same one as core oral microbiome. Okay, move to this slide. There were several studies about oral microbiome worldwide. So I would like to show you an example of the study. This study 120 individual saliva worldwide, and the result shown that no significant geographical difference, no diet or environment factor that influence the composition of oral microbiome. So it means that the genetics or host species may be influence the composition of oral microbiome. So they said that something that produced or released together with saliva and gingival cervical fluid, mostly the acid, can feed the bacteria. So different in these factors might affect the composition of the bacteria. So it seems that if these factors promote the good bacteria, so you will get a healthy oral cavity. But if these factors feed or provide the availability of the bad bacteria, you will get an unhealthy oral cavities. Okay, in this slide, show the species, the nomenclature at the species level of the important and predominant oral microbiome. So it can be classified into the above one, the one that have high composition in the saliva. And among them, there are three genera have important for health. For example, Porphyromonas, especially the species Porphyromonas gingivalis. Okay, when moved to the higher level or the phylum level, so list here are the phylum that found almost 96% of the species detected. But the important phyla is Fusobacteria. So you will hear later about Fusobacterium nucleatum. Okay, next. At this point, uh, should we try to answer this question? Why most non-oral bacteria are not routinely isolated from the mouth? This means that why we could not see other bacteria that normally found at the other side on the oral cavity. Okay, the answer is that the other bacteria or non-oral bacteria may lack adhesin and receptor. Adhesin is the surface protein of the bacteria that can adhere or stick to other environment. It might be the other bacteria or on the enamel of the teeth itself, as well as the receptor. So these adhesin and receptor would be able to would enable them to bind the oral surface or coagulate with other bacteria. And the second answer or reason is that there may also be immune exclusion. It means that some antibodies that are secretory antibody can exclude or destroy the other bacteria or eliminate the bacteria from the oral cavity. Gut coliform are rarely found in health but often found in immunocompromised individuals and those with reduced salivary flow. It seems that some bacteria that coliform, most coliform bacteria are found in the colon, right, in the feces. But sometimes it can be found on the oral cavity. So the reason is that the situation we found mostly in the immunocompromised individual or individual has, reduced salivary flow or they have a low amount of saliva. So it seems that immune in the healthy people can exclude the other bacteria from the oral cavity. Okay, from now we will talk about the role of microbiome in health, or we try to see the benefit of the oral microbiome on health of people. Okay, for healthy oral microbiome, it is the collection of the good flora or commensal microbiota. So the bacteria that present in the oral cavity but has no impact to pathogenesis of any disease. So development of local and systemic immunity, immunity on the oral cavity is activated by the presence of the oral microbiome. They found that the lymphoid follicle will not develop in the germ-free mice. The mice that grown without any presence of the microorganism in the environment could not develop the immune system properly. As you will see, the lymphoid follicle won't develop because the microbiota can activate mucosal immunoglobulin A to to release from the macrophage and destroy or control the population of the bacteria on the oral cavity. So if you alter or disturb the oral microbiome, especially the good one, with antibiotics, it might alter the, I mean, symbiosis of the bacteria. So some of the commensal microbiota will dominate. For example, the Candida species, it will dominate, or Staphylococcus aureus, especially in the HIV infected individual, will dominate. Okay, move to this slide. This is an example of uh, the mechanism and the link between the nitrate metabolism that produced from the oral microbiome to cardiovascular health. Nitrate from food will change to nitrite. Nitrate is NO3 minus, but nitrite NO2 minus, as you can see in the figure. Nitrite will produce from the oral microbiome and circulate between enterosalivary circuit, and some of them will release into the bloodstream. And the nitrate is good, or it is benefit to the health because it will stimulate the vascular health, especially vasodilation. So it will reduce the blood pressure. It seems that if you have a good oral microbiome, it will link to the normal blood pressure. So in contrast, if you have a bad oral microbiome, it will uh, trigger the high blood pressure because the body lack of nitric oxide that will be produced from a healthy oral microbiome. So it might be some factor that as a public health student you might use oral health to determine the healthy of the elderly, especially elderly with hypertension. Okay, apart from the healthy oral microbiome, we move to the disease that can be caused by oral microbiome. In this case, I will say that it will be unhealthy oral microbiome. Okay, let's start with the dental caries or tooth decay. As I previously mentioned about the acid on the oral health, acid is a product from fermentation of carbohydrate by oral bacteria. This acid will change the buffering capacity of the saliva. So if the pH or buffering capacity of saliva change, it will change oral microbiota as well. S. mutant and lactobacilli are the group of bacteria that produce acid. And the other bacteria that found in the plaque biofilm that also be able to produce acid are bifidobacterium, propionibacterium, and Saccharomyces. Okay, actually the basis or base that produce from the other group of bacteria will balance the acid. So it seems that the bad bacteria produce acid, but the good bacteria produce basic. Instead of ferment the carbohydrate, the good bacteria ferment urea or arginine to ammonia to increase the pH of the oral cavity and saliva. Okay, the second, the unhealthy oral microbiome for endodontic infection. So the cause of endodontic infection is by untreated dental caries lesion, and most of the bacteria that can survive in that condition in the deep uh, deep area like these are anaerobic proteolytic bacteria, for example, enterococci, which is uncommon in the mouth. It's mostly found in cheese, in both pasteurized and non-pasteurized. And none of the other normal flora on the oral cavity can complete these enterococci if it localized in the root canal. So the patient that receiving the endodontic treatment should be advised to avoid the consumption of the food that contain the enterococci, as I mentioned before, which is cheese and both pasteurized and non-pasteurized form. Okay, move to the third one, periodontal diseases. So before going on the sub detail about the periodontal disease, I would like to tell you about environmental and endogenous factors that are associated with the increased incidence of periodontitis, which include oral hygiene, smoking, obesity, stress, and potentially genetic association. Start with a subset of periodontal disease. The first one, gingivitis, which is the most common, almost 90% in adult. It is a result from biofilm formation after dental plaques. There is an event that occurs after the presence of dental plaques is multi-species aggregation of the oral microbiome. The salivary glycoprotein that produced from the bacteria will be adsorbed into the teeth to form salivary pellicle. After that, there will be two groups of bacteria that will be co-absorbed into the salivary particle. Yes. It will finally produce the biofilm, sometimes you call a co-aggregation of the bacteria. As I mentioned, there are two groups. And the first group are primary colonizer. Mostly they are gram-positive arrow and some facultative N arrow. For example, streptococci and actinomyces species. And the second group are anaerobic organism. It will help to produce the mature plaque, which are gram-negative, such as Fusobacterium, Treponema, and member of the phylum of Synergitis. As you have learned from this course, gingivitis and the inflammation of the gum is the reversible event. It means we can cure it because it's the starting point for the next one, the very severe inflammation. Okay, the more severe one, or periodontitis. This disease is not reversible. We cannot cure it. So it is the permanent damage of the bone and the gum. So at the periodontal pocket is heavily colonized with anaerobic bacteria, and the host response and host immune response will trigger the release of protease that make a significant contribution to the tissue damage. A specific bacteria, a specific microbiota is associated with the advanced periodontitis, but this event is unclear. Some literature report some species, for example, Porphyromonas gingivalis, Trypanosoma denticola, and Tartanella forsythia. However, it seems that interaction between the unhealthy oral microbiome with the host response will trigger this event, this situation. However, if the patient get some strong environmental factor, which is tobacco smoking, it will enhance this event to occur sooner. Okay, in this figure, it is the diagram showing the difference at the tooth, the surface, the junction between the gum and the tooth of the mice that grown with and without the bacteria. If you pause this video, you will see the difference between the germ-free and conventional. So in the conventional, mice growing in the presence of bacteria, you will see the biofilm. The additional is the biofilm present when compared to the germ-free mice. So in the biofilm, you will see a group of bacteria, several bacteria, as mentioned, primary and secondary bacteria. The gram-positive and gram-negative one, they cluster together, and you will see the PMN or white blood cell that sometimes it might cluster with the biofilm. So this is the difference between the mice that grown with and without the oral microbiome. In the oral microbiome present mice, it will show the biofilm as you can see in the figure. Okay, move to this slide. This slide is the same story with the previous slide, but it shows you more detail. So we have to compare again between germ-free and conventional mice. So look at the first thing that are different between these two types of mice grown with different conditions. In the mucus layer, in the conventional grown mice have thicker, and you see the capillary system that supply to the mucus layer or supply to the cell at the surface area have higher and more, it has more complexity. As you can see, again, it has a lot of immunoglobulin A in the purple. Right? It has more immunoglobulin A secreted from the conventional grown mice because it has a lot of blood cells that supply to that area. So these are the difference between, the healthy of the epithelial cell that close to the teeth at the oral cavity of the two different grown mice, the germ-free and the conventional mice. So conventional grown mice seem to have the higher, amount, volume of the mucus layer, capillary system, and the blood cell that supply, including the antibody that supply to that area. So it seems that with the presence of oral microbiome, it can trigger or promote the healthy of the oral cavity, especially at the epithelial area when compared to the germ-free. Okay, the last topic. Sometimes we can observe the unhealthy oral microbiome among the people suffer from non-oral disease. For example, disease from infectious endocarditis, the abscesses. It may be from brain or the liver, cystic fibrosis, and the last one, soft tissue infection. So I would like you to let you some question that oral microbiome can be developed to be a biomarker for other disease. This might be you can link the oral microbiome to the other disease. Can it possible? Okay, next I would like you to understand the concept from this topic. So oral microbiome, it has their characteristic different on several factors, but mostly it depends on the host factor. Okay, the interaction, look at this triangle. So the disease will develop, it depends on the association between these three factors. The first one is the proportion of the oral microbiome. So if it can regulate that this proportion that's suitable for everyone, or we call symbiosis, it will, help to protect other factor that will lead to the disease. Or sometimes if you have a lot of violent factor, for example smoking, it will disturb this balance of the the triangle. Sometimes getting parasitic or other unbalanced microorganism may come from parasitic or opportunistic relationship of other microbe that will disturb this balance, will also lead to the disease as well. And for this slide, it shows possible mechanism which the inflammation may modulate systemic disease. For example, the inflammation will trigger the releasing the bacteria from the gum or from the oral cavity into the bloodstream, so we call bacteremia. And the content of the bacteria, especially in the gram-negative bacteria, it has the LPS or lipopolysaccharide. This will trigger the immune response that you will feel it as you will get the fever. Or the inflammation will trigger the inflammatory modulator, some protein that release, it might be released from our white blood cell, for example IL-1, IL-6, and TNF-alpha. This also trigger some or other organism, or other organ, for example liver. And the other pathway is the immune response that respond to particular species of bacteria, that this response might affect to other organ as well. Okay, lastly, I would like to show you how to study oral microbiome. This is just one example of study. This study, they use the technique to analysis of diversity of the bacteria by using the TRFLP technique. This is the tool that used to monitor the diversity of bacteria. The subject in this study are 200 Japanese, the age between 15 to 40 years old. So they use sample as saliva. They use the advanced statistics to classify the data of oral microbiome composition. They can classify into three groups: group 1, group 2, and group 3. And look at the table 1. In the table 1, they try to describe the parameter of interest, for example, the teeth condition, parental condition, and you will see that there are some significant difference among these three groups. For example, the especially on the periodontal condition. So it seems that the bacteria might, by using the bacteria composition, we can classify them into different group, and they show the difference in periodontal condition. Okay, on the last slide, I would like you to see how they analyze the composition of the bacteria on oral cavity from the participant, as they classify them into three groups. Look at the first graph on the left hand side. You will see that there are three clusters, cluster 1, 2, and 3. They differentiate, or they can be, distribute and cluster into the closely related group 1, group 2, group 3. As you can see, the analysis for this figure, we use principal component analysis, which is the advanced statistics. And on the right hand side, they classify by relative proportion of 6 dominant bacterial genera. So you see the different proportion. It seems that on the cluster number 2, it has a lot of streptococcus, but for cluster 1, it has a lot of the Veillonella, and the cluster 3, it has a lot of Neisseria. Okay, this is an example of the study about oral microbiome, and they can use the diversity of the oral microbiome to link with the periodontal condition, as you can see in the previous picture. Okay, so this all I would like you to know about these topics, and I would like to thank all students that you have learned my topic by using my video clip. Okay, good luck every student.